So this is an important slide to understand what we were thinking when we designed this trial. And it's important so that you understand the subsequent information that I present. Our goal was an efficient, rapid development program, despite the chronic, slowly progressive nature of this disease. Placebo groups in this disease class transition very slowly. They decline slowly. There have been a number of negative studies targeting groups mild, moderate Alzheimer's disease with disease-modifying drugs, and they didn't all use enhancement approaches such as PET or CSF biomarkers to make sure the patients all had amyloid-positive Alzheimer's. So, our considerations shifting to an earlier group of patients, early Alzheimer's disease, rather than mild to moderate. Obtain strong clinical proof of concept. In other words, with a phase 2b study, you don't want to make decisions too quickly. You want adequate information to decide you're going to invest these huge sums of money. We needed to evaluate dose-ranging effects, so we wanted multiple doses and enough patients so that we could look at subgroups. We wanted to account for the duration of the study that's required for disease modification, 18 or 24 months. We wanted to account for the magnitude expected of a treatment effect, given this slow decline. And we wanted the potential for early decision-making and the 12-month endpoint, which I'll touch on further, while preserving the 18-month potential for disease modification. To do that, we needed some innovation. And so we looked at two different things. One was this clinical response-driven adaptive randomization. And I will talk more about that later because it's very important to understand how well that worked in this trial. Secondly, we needed an endpoint that was sensitive in this population to allow the adaptive randomization to have its full effect. And I'll discuss how we developed this ADCOMS later. So, the population. This was an early Alzheimer's population. It was amyloid positive by PET or CSF, MMSEs of 22 to 30, and CDRs of 0.5 or 1. The key design components, it was 18 months duration and approximately 800 patients. That was our target. Treatment of 6 arms, 1 placebo, 5 active arms, 2.5 milligrams biweekly, that's twice a month, 5 milligrams monthly, 5 milligrams bi-monthly, 10 milligrams monthly, and 10 milligrams bi-monthly, so 5 groups. Important thing to know here is that all subjects remained on the same randomized dose throughout the study. In other words, if you were randomized to 2.5 milligrams, you stayed on 2.5 milligrams the entire time, and all subjects received biweekly infusions. In other words, if a patient was randomized to 5 milligrams once a month, the second infusion was a placebo in order to remain blinded across the study. And that's very important for the integrity of the study. Next, we use periodic interim analyses to allow the Bayesian allocation to take effect at its fullest. Now, how did we do that? This was a pre-programmed, computer-generated algorithm that allocates patient based on the probability that the computer sees that that dose might work in the end. And I'll describe that a little more, but it sounds complex, but it's really not so complex. What were our primary endpoint? ADCOMS. My next slide, I'll discuss that in detail. But we also have ADCOMS, ADAS-COG, CDR sum of boxes. We had 2 sub-studies, PET and CSF, And we also had a volumetric MRI. We don't have those results yet, and I won't be describing them. Safety. The safety, all area E was required to discontinue per protocol, even if the patients weren't symptomatic. And I'll describe that on a subsequent slide so you understand that. That's very important. So, ADCOMS. As I described, in order for this allocation to work well, we needed a very sensitive endpoint that would allow allocation to occur early in the course of the study. So we thought, how might we do that? We took 5 validated systems, and I list them there. The 3 most important turned out to be ADAS-Cog, MMSE, and CDR sum of boxes, well validated in mild to moderate populations. Keep in mind, Alzheimer's research has been going on for the last 20 years using those. But in those days, we didn't study prodromal patients. Nobody was quite sure what a prodromal patient was, what an MCI patient was. So these scales were developed to assess patients that were more significantly impaired than the population we would look at here. So they couldn't be as sensitive as we would need to cause an allocation early. So we had to do something with a little bit of innovation. So we took 4 study databases in MCI patients and 4 study databases with mild AD. These both groups included the NIH ADNI longitudinal cohort patients, and that was the core of this analysis, to develop the elements of our ADCOMS. The elements of the ADCOMS are 6 domains from the 6 domains CDR sum of boxes, 4 domains from ADAS-Cog, and 2 domains from MMSE, all components of existing scales. These scales were evaluated in computer modeling to identify which were the most sensitive in the population we were trying to study, the milder patients. And we evaluated the performance of those, comparing it with traditional endpoints in an article that's listed at the bottom, Wang et Logovinsky et al. And those had at least 20% greater sensitivity in this patient population. We've replicated that, or we had some assistance from Roche who've replicated it. They took their Scarlet Road study, presenting that a couple of years ago at the ADPD, where they showed that they also found about a 30% greater sensitivity using this scale retrospectively in their analysis comparing to the traditional scales. We have a poster on Ellen Bessestat, which was presented at 1 o'clock today, that replicated that, and we replicated it again in this study. So I'll show you that in just a moment. So these are the endpoints in the middle, secondary endpoints, but they're the 12 and 18-month final analysis. We've already explained the primary endpoint at the interim analysis at 12 months through a Bayesian analysis. And I'll give you an idea why, what kind of a hurdle. It was a very high hurdle because we didn't want to make a mistake. And I'll explain that with some examples that should help you understand that. We're going to present data on baseline PET and SUVR amyloid loads, the conversion of amyloid positive to amyloid negative, change of baseline ADCOMS, ADAS-Cog, CDR sum of boxes, and CSF data, A-beta 1 to 42, and total tau. You're going to see slides with that data in just a second. But first, let's talk about the Bayesian allocation. It was supposed to identify the top 2 most meaningful doses and allocate most of the new patients to those doses. And as you can see, out of the 856 patients, the top 2 doses were received 414 patients. Placebo was always targeting approximately 250 patients. We had 247.